Hello friends, welcome to lecture 3 of week 10. So in this lecture we will understand what are the applications of different multiomics approaches that we have discussed so far, and particularly their application in terms of human health, disease, how they can be used to diagnose the disease or assess the health status or to suggest therapeutics, right? And we will also study some examples of cancer, diabetes, where such kind of multi-omics approaches have been used successfully to understand the disease, its progression, diagnosis, and also for the therapeutics. So before that, basically the most important achievement to deal with human health was the beginning of the human genome that began almost in 1995, and the first draft came in 2001, where basically the good draft level sequence of all the human chromosome was available. So while this was going on, the chimpanzee genome and other primate genomes was also going on, which is actually very important for the comparative genomics to see when we are dealing with different diseases, because it could be that in chimpanzee it may not show the disease, but human can. So basically this comparative genomics also we can get from there. So while talking about the human genome, it was not only the genome, it led to the transcriptome, metagenome of the human body, epigenome, right? And also like the proteome. So basically all the different type of omics evolved with the while this human genome was going on, and the final sequence of the human genome appeared in 2022, where it took almost 32 years to complete, and it could give the telomere to telomere, like from start to end coverage of the human genome. So people were dealing with different drafts of various good quality, but since there was still some gaps in the human genome, and now since we have established a benchmark in terms of human genome, the genomics of other organisms and its importance in the health of those organisms or to the environment, right, can be studied. So now when we have the human genome, it provides an opportunity to carry out the genome-wide association studies, which are very important to understand which loci in the genome or which regions in the genome are basically correlated with the disease, right? So people do these as kind of association studies with different type of diseases, and the best examples are the diabetes and cancer, and right. So in these mostly these regions are the SNPs, single nucleotide polymorphisms, which are basically at any given position the 4 DNA nucleotides can vary between the individuals, and this variation also is not that any 4 can exist. Basically purine can replace a purine, that means A to G, or pyrimidines to pyrimidines like C to T. So this kind of variations are more common, transitions within transversions, right? And through these studies, people have been associating diseases with various locations in the genome, and this can help in pre-diagnosis or basically like telling that okay, you are predisposed to a disease, or basically there are a lot of SNP arrays and sequencing based approaches also available for that uses these data. So basically it is like you can select a number of individuals and collect the genomic data from that. Then you can do the genome typing to see that at any given position in the genome, what are the sequence variants that you observe in populations. So it is better also like sometimes ethnic groups or different population groups can show different some differences in their genomes, right? So nowadays with the availability of more and more data, you can do the regional or geographical studies based on the genomes available in like different regions, like representatives from Asia, Europe, Africa, or America. And then for the different persons, you can look at the SNPs, the sites which are varying in the different individuals, right? Of which some sites could be more important and that show a consistent variation across individuals, right? So if in the two nucleotide that may occur at those location, you may find that the presence of one nucleotide is actually associating with the disease, okay, replacement of say T by G and like that, okay. And then you test these associations, do the statistical analysis in different cohorts. You can take from multiple population like this, it is shown here, the color coding is also there, right? And then once you have established these markers, you can now expand this on another subset of population or a different population and see that whether that holds, and do the post genome wide citizen studies analysis. So basically these studies have been very important to find like thousands of variants across the genomes, and particularly like in case of diabetes, people have found that basically almost 50 genetic loci are associated with various glycemic traits and 90 loci with type 2 diabetes. Okay, so we'll see the type 2 diabetes in the next slides, but this gives an example that there could be 50 or 90 different locations across the genomes and spread across the chromosomes, maybe in the genic or intergenic or intragenic regions, right, or in the coding regions, which are actually responsible or associated with the disease traits, okay, or the phenotypes, okay. So basically this can be helpful in explaining as much as 10% of the variance in disease susceptibility, right? But it is not that presence of these markers will definitely predispose a person, but these can provide very important clues about the predisposition, okay. And there are basically, diabetes is multifactorial, and so many environmental and external factors or internal factors or the other comorbidities or diseases can actually also act in together to lead to diabetes. So but this GWAS is also one of the important tool to study it. Then while we are doing this, you can also see the comparative studies in different animals. You can even use the animal models to test your disease. So we always choose the animal model where we can actually replicate the disease, and which can be a good model to see the disease phenotype, and also trying the therapeutics. So these kind of studies are also a part of these omics, basically the multi-omic studies that are aimed to study various diseases. Then another important thing that we have seen, that along with the GWAS maps or the genomic map or the microsatellite maps, there is also epigenomic maps that have been generated for the regions. Basically these are the CPG islands or methylated Cs across the genome, or at different locations in the genome, which when are methylated, they can actually repress or activate the suppression or expression of a gene, okay, which we can actually determine by the bisulfite sequencing or by the next generation sequencing, including Illumina or PacBio hi-fi reads. But if you see like almost like in these CPG islands or this are also located across the genome, and it is also not and their location has been correlated with various diseases, the presence of these CPG, right? So people have tried to construct the epigenome map, and like this epigenomics program is there, which creates almost 1000 epigenomic maps per year. Another important aspect is the single genomics and human health, because either you can study the entire tissue, for example, you take blood from an individual or some tissue or scraping or some cancer tissue, okay, so you can extract the total RNA from that tissue, okay, or total DNA from that tissue, but that is giving you a profile for the tissue but not for the single cells, which actually could have say different level of gene expression or mutations or epigenetic modifications which can be associated with the disease. So that is why basically this single cell genomics offer to understand the disease pathogenesis better, because then you can understand the cell to cell communications, like how the cells are communicating, what metabolites or signaling molecules they are producing to basically act in accordance to the other cells or differently from the other cells in their respective microenvironment. So that is a spatial analysis, how they are interacting with the other cells in that, and also this can also be used to understand that how the cells are dividing and how these epigenetic modification may change on those. Okay, so epigenome, single cell genomics. Okay, and also like single cell genomics has an application in understanding the specific cell expression, gene expression, metabolomics of that cell, or even the immunoprofiling of that cell. So this we have dealt in the previous lecture. So we also discussed earlier that how we can actually separate the cells to prepare the single cells. So basically FACS or fluorescent activated cell cytometry is one of the very important method. Then laser capture microextraction can also be used. So these things we have discussed in the previous class, so I am just giving you an overview and connecting with the current lecture. Okay, similarly like microfluidics that we do in the case of the chromium instrument or the micromanipulator, where we can handpick the cells, separate the cells that look different under the microscope, and take those cells and then rupture those cells and do the genomics. So these are the methods that can be used to actually get these single cells for various studies. And once you have got this, basically not only health, you can also understand the disease patterns, like in a tumor also, like there are cells that are heterogeneous and very different than the normal cells, and even in the cancerous cell there could be a lot of heterogeneity, so that can be understood by single cell genomics. Again, like you can do using those cells, you can also do like responder versus non-responder studies of any kind of treatment that you are giving, that can provide clues for the different drugs combination that you can provide to those single cells, which shows differential phenotypes, or to understand the various cell to cell interaction in that tumor microenvironment where the cells have been heterogeneous. So basically this single cell genomics today has appeared a very important tool to understand the disease progression, diagnostics, and also for giving the personalized treatments. Like we can understand an organism in terms of the cell, which actually form the tissue, and tissues form the entire organism. So we can have profiles for multi-omics profile. It could be genomics, transcriptomics, proteomics, metabolomics, epigenomics. Okay, for the healthy tissue, for the healthy cells, for the disease cells, and estimate the disease progression and what changes happen in the respective omics analysis of the different single cells, which can provide clues for the personalized treatment. And basically today these studies are also aimed at multiple omics analysis, so that any disease or any condition or treatment response, anything can be studied in more detail by covering all the 4 major omics components. Similarly, like transcriptomics is very important to understand the disease, and there are so many thousands of studies that shows the differential level of transcripts when measured in the healthy and disease conditions, or between twins. People have shown that some genes are varying, and it is not that all the genes show same expression, cancer versus normal cell. And basically transcriptomics can provide the first hand information about how the cell is responding to its environment, and how the transcripts are varying with response to the microenvironment or to some external factor. So basically this transcriptomics is nothing but you are studying the entire set of transcripts in a cell at that point of time, because at another point of time, like you are sitting, running, eating food, before food, after food, even different stages of life, this transcriptome may change, disease treatment, sleeping. Okay, so basically you have to capture that transcriptome for that moment, and by extracting the total RNA from the cell, and then you are more interested in the mRNA that forms the functional proteins, and you get rid of the remaining structural RNA, for example the ribosomal RNA by ribodilution step, so that otherwise if you sequence or do any kind of analysis, there will be a lot of contamination. So basically you are dealing with the mRNA component in the transcription, in the transcripts, and this helps to understand the disease expression level that vary during the disease, or regulation of genes in various biological systems. You can conduct a high throughput sequencing analysis, because by sequencing you can know the copy number as well as their expression information, like which genes are expressing and how much they expressed. Okay, correctly you can count each and every transcript as an evidence. Whereas in terms of microarray, which we discussed earlier, there you can get an indirect evidence of that the gene is expressing, and how much fold difference it is having in various disease conditions. So microarray and RNA sequencing are two important techniques to study the transcriptome of an organism. So basically you have to identify the organism and do the total RNA extraction, and then basically purify the mRNA by getting rid of the other types of RNA in the cell, fragment it, convert into cDNA, because mRNA cannot be sequenced because it is very unstable also. So you convert that into cDNA, and then by ligating adapters at both ends, and or following the protocol which the sequencing library provides, or the sequencer requirement is. So you can construct the library, and since you may require sufficient copy number of each DNA fragment, so this clonal amplification step is also needed, for example in Illumina instrument, but it is not a requirement in the nanopore or PacBio. Then the raw data that you generate after the sequencing, you carry out the analysis, and then basically you relate this data with the genomic data, with the GWS data, epigenome data, or any other kind of, like we will also see the metagenome, metabolome, and different omes data you combine together and complete the story about the question that you asked about the disease, or therapeutics or diagnosis. And some tools are there we have discussed earlier, like Trinity, HiSite2, CD, EST, which can help in the transcriptome analysis. And basically depending on the context and what you really want to do in transcriptome analysis, if you search you can find so many tools that are published. This is an established field, and so many publicly available tools are available for transcriptome analysis. Then again, like in transcriptome, single cell gene expression also matters, because in the previous I showed that you are extracting the total RNA from multiple tissues, because you need sufficient quantity of mRNA. But from a single cell also, you can, using the Chromium instrument, you can basically separate the cells from each cell which is attached to a bead. You can actually amplify the RNA for that cell and go for the sequencing, so which can provide information about the expression of genes in that particular cell. So it works like this, like you may have a tissue, it could be solid tissue, cancerous or normal healthy cells. Okay, you separate the cells, then once you have the single cell status, you can isolate the RNA, particularly the mRNA, convert that into cDNA and then go for the one round of amplification, so that you are having sufficient quantity of RNA, and then followed by library preparation, sequencing, and the single cell expression profile from the multiple cells you have got, which can tell you a lot of clues about how the cellular expression varies, and in terms of the tissue in comparison to the tissue expression, or any health status, or any treatment response, or the question that you've asked. Now, in addition to single cell genomics, today one of the major areas also spatial transcriptomics, where you can because in single cell also you are taking out the cell from its respective environment and doing the analysis, the transcriptome analysis, and that may change the expression level, because the cell has undergone some kind of stress when you are taking out that cell from its environment. So in spatial transcriptomics, you can measure the transcriptome or assess the transcripts of all the cells in their respective environments. So which is very good to see the real-time picture of how the gene expression changes are actually leading to in the different cells in that particular tissue, and basically leading to disease or how they are communicating. So you can get a direct evidence from there. So the most of the things are similar, like for example, in this case you have an array of spatially barcoded mRNA capturing spatially barcoded and plus mRNA capturing plus reverse transcription primers. So each of these array contains that, and each array is basically contains, each individual spot like this contains millions of copies of an individual primer, okay, which looks like this, which contains mRNA capture to capture the mRNA from the tissue, basically cell in that tissue, the barcode for that particular tissue, that means all the components DNA or RNA that you are extracting, and reverse transcription primer, right? So basically this will attach to the different cells like this, and will get the contents from each and every cell, and then in this array you can get the differential expression of genes in different tissues during the in their normal condition. And this has a lot of applications, like tissue homeostasis is basically at a healthy for a healthy tissue at single point of time, what is the the homeostasis, that is the tissue's homeostasis, and but it may change because of various conditions or treatments or time points, right? Or so basically you can study from these single cell RNA-seq and spatial transcriptomic data can tell you about the tissue homeostasis, how the tissue developmental level of the tissue, like the development during the developmental phases, how the tissue expression profiles change in the different cells, right? The microenvironment of each and every cell, and how that changes the gene expression level, and similarly like disease microenvironment also in terms of proximity to amyloid plaques, etc. So this transcriptome along with genome, metabolome, proteome is very important area to completely understand the disease, and along like people are doing things on large scale, like you have the human epigenome map or genome map, microsatellite map, or okay, similarly you have the human spatial atlas also, where the spatial transcriptome data from different tissues have been deposited, and people are depositing it there so that it is available for global research. Then human microbiome is recently emerged a very important factor for health and disease. So microbiome is the you can say this bacterial but like typically the bacteria that are harbored inside the body or on the body, right, in the gut or on the skin, but it may also contain the fungi, protozoa, viruses, anything which is microscopic. So that also constitutes the microbiome. But people generally, when we are referring to microbiome, they are referring to bacteria that occupy these niche areas. So if you say the total number of human cells in the body, so basically the number of bacterial cells actually outnumber the number of human cells, and you have bacteria all over the body, including your gut, skin, scalp, armpits, or various other sites. And basically today it is known as our second genome. And for example, in the gut you have almost 2 kilograms of bacteria, more than 1,000 species. And when you say I have a gut feeling, basically we're actually talking about the feeling of those thousands of millions of bacteria inside your gut, which are actually helping in degrading your food, fending off the pathogenic microbes, protecting the epithelial cells, stimulating the immune system, and also metabolizing whatever you are taking in. Okay, so basically in the large intestine they play a very important role. Similarly, like skin is a very exposed organ. You are dealing with environment, the toxins, the pollutants all the time, and the bacteria are kind of sticking on your skin and doing some function, like in the scalp or on the skin. Oral cavity, when you are eating food, basically bacteria is there in the teeth, or forming biofilms, or inside the oral cavity, which are also doing their own job. So basically this environment is very, very important for human health, and when the dysbiosis happens, it causes various diseases. And most of these bacteria, because of their environment issue, and we do not understand that their requirement for culturing, they cannot be cultured. But today, like, people are doing a lot of efforts to culturing them, and but still you can say that almost 98% bacteria on this planet cannot be cultured. And that is why metagenomics is very, very important, to directly sequence the bacteria and their respective environments without the need of culturing or further amplifying them. So that is the only way you can access the genetic material. And to talk about the gut microbiome, which is one of the very, very important microbiomes for the human health. So India is a large country, and people have been doing the gut microbiome metagenome from various populations. So when we say microbiome or metagenome, the difference is that in metagenome you're talking about the circular chromosome, and using the shotgun approach to break down that circular chromosome and sequence everything in that environment, all the genomes. But in case of microbiome, when you say it, is mostly you are referring to some amplicon based or marker based approach, where you can identify the microbes and also their respective numbers, but do not get much details about their functional components. So basically people are generating the microbiome metagenome data from all over the world, different populations, and like European, African, American, Asian, Indian subcontinent, or Chinese, Japanese. So many microbiomes are available, but there is a lot of diversity. And even if, like, consider the case of India, there are several geographical regions, ethnic groups, socioeconomic differences between the people, lifestyle differences, right? Food habit is different. Whenever you cross borders, your diet is different, age difference is there, veg, non-veg different. So many factors impact the net gut microbiome, but still there exists a core gut microbiome which is important, which is basically the marker for that population, and also tells you that how those microbes in their healthy state is contributing for the human health. So basically I'm just referring to one study which took samples from different geographical regions, and also could do the comparative analysis with the other population to understand that how this population of India is different than the other populations, and how the healthy population also microbiome differs from the disease microbiome, such as the IBD or Crohn's disease in Western populations. So the methodology here would be like the metagenomic approach, including both amplicon based and shotgun based sequencing. And in metabolome, you can, because you are using fecal matter as the the tissue from which you are extracting the DNA, because you want to get a glimpse of the large intestine microbiome. So basically this fecal microbiome also contains the metabolites, which is the end product of the metabolism of whatever has been eaten, and also the action of microbes. So fecal metabolome is important that way. And serum metabolome, because the metabolites which are absorbed by the host, they are in the serum. So basically studying them can help you to correlate the action of microbes, role of host, what has it absorbed and what is excreted out, and tells you the total picture of about the metabolism in a healthy, and also when you compare with a diseased individual. And this is the paper that that referred to this research. And other important finding from this emerged that Prevotella copri emerged as the most abundant bacteria in the Indian gut microbiome, which is almost like 30 to 90% in of the gut could be this particular species. And however, the other species of Prevotella were found in the Western populations, and they were mostly the inflammatory species, whereas the Prevotella copri, though sometimes have been associated with some diseases, but it is found in the healthy human gut microbiome in the Indian populations. And actually this is contributing a lot in the polysaccharide degradation of because Indians diet contains a lot of carbohydrates and polysaccharides, plant based polysaccharides. So we need bacteria and their enzymes to metabolize those, because those are not present in the human. And Copri is doing that job in the Indian population along with some other microbes, whereas the Bacteroides is doing the job in the Western population. And that is why basically studies can do like more exhaustive search for that particular bacteria like Protella copri and build their respective database, associate that with disease and healthy status in the different populations. For example, in this picture you can see that the differential distribution of Protella genus across the globe, and the population that were considered, that how it varies. For example, you can see that in the African and Indian populations, Protella is the main bacteria, whereas in the other US, Netherlands, the Bacteroides, Ruminococcus are the key bacteria. Okay, so and basically you can see clear differences between the genome that are present in Western and non-Western gut microbiomes, which is very important for health, because when you're talking about probiotics or any diagnosis or therapeutics, so you have to assess the individual or population specific gut microbiomes to do more kind of personalized treatment or diagnosis. And also, like broadly, it showed that basically the Indian gut microbiome is very different from the other populations, and this big circle means that basically there is a lot of spread. That means even in India there is a lot of diversity in the gut microbiome. Still, Protella was the most important microbiome that was observed. And then, like on detailed examination of the role of Protella copri in the gut microbiome, it was observed that it actually contains the polysaccharide utilization loci in its genome, which helps in the degradation of various plant-based materials like various it contains basically the enzymes for degradation of those plant polysaccharides and carbohydrates, which basically a database such as KZ, carbohydrate active enzymes database, can be a very good database to access the enzymes which are, because they have already categorized the various carbohydrate metabolizing enzymes, and now we can map the data from any population on that database to understand that which particular enzymes are more or less in that population. And it found that basically the clusters are present, polysaccharide utilization loci clusters are present in different Protella genomes, and which are actually contributing to the polysaccharide degradation in these populations, in the non-Western populations. And while comparing that with the, so just to give you like idea about how it is can be associated with the disease. So basically, Protella copri was found to be more abundant in non-Western, including India and other African populations, whereas the other species of Protella were abundant in the Western gut microbiome, and those have been associated with various inflammatory diseases also, such as IBD or CD. And it was also noted that those inflammatory or sometimes also called pathogenic Protella other species were also found in the mouth of the Western populations, which indicates or gives you some clues about the mouth gut axis, that bacteria from the mouth can go to the gut and colonize the gut later. So basically that means the oral hygiene and the gut, basically this is there is a crosstalk. And also, like people have understood the gut brain axis crosstalk, that how the gut microbiome can influence the brain. Similarly, like mouth microbiome can also influence the gut microbiome. So basically this kind of study provides evidence or very good insights about how the variation in diet can lead to differential gut microbiomes, and how that varies in the healthy and diseased individuals, right? And what is the role of diet in changing that microbiome. And so there are many insights were there. So basically this study or basically the understanding of human microbiome in gut or other body sites also is very important. That is why for the human health. So this is one of the omics which is coming up very nicely. For example, now if you move to the cancer gut metagenome, you can see that when you compare the cancer and healthy gut metagenomes, you can see that there are some distinct genes and taxonomic markers, that means the bacteria which can separate the healthy and diseased cancer versus the healthy gut microbiome. And this kind of finding can provide clues about using them as markers for early diagnostics, or because this will also be a non-invasive diagnostics, and colorectal cancer is a very big problem, almost second leading cause of cancer deaths. Although in India it is less, but it is increasing because of the Western diet. And if you can early diagnose this using the feces, which is without actually going for colonoscopy or biopsy, right? So you can know only by the feces, by assessing these markers, that whether there is a possibility or what stage, because for example, this has the 4 stages, so you can detect in any of the 4 stages of the cancer. And so basically microbiome, along with some data analysis using AML based approaches, can be used for diagnosing cancer in a very early stage. Similarly, like autistic gut microbiome, because the autistic children have issues with have a lot of gut related problems, and microbiome could be one of the factors for that. So when the gut microbiome of healthy and diseased children were compared, so it was found in India basically. So basically they were also on the normal diet, not on the gluten free diet. So it was noted that some bacteria were highly overrepresented in the gut microbiome of the autistic children, which gives clues about how these overabundance of microbes could be associated with the syndrome. Okay, but it could be the cause or effect relationship that we still do not understand, that because of autism the microbiome changed, or because of the change in microbiome dysbiosis, basically it played some role in the autistic syndrome. Then moving on to the scalp, which contains basically the fungi and bacteria, and we often find dandruff situation and healthy scalp microbiome. So all the studies were done so far in the Western countries. So one study was performed in the Indian context also, in coordination with L'Oreal, where basically the scalp, so how basically from the scalp you can get the sample. So you can for the previous one, like feces, you can get sample in the small containers and transport that to the lab at in the freezing condition -20, or immediately process. Or for this scalp microbiome, you can use swabs, okay, and then extract the DNA from that, the bacterial DNA and fungal DNA. So for which basically the bacterial and fungal DNA extraction, amplicon and metagenome sequencing was performed, or for the fungi it was ITS and metagenome sequencing, which can provide clues about the differences in the healthy and dandruff scalp. And basically the study found that the proportion of the bacteria, basically the healthy and dandruff scalp can help both kind of the healthy and disease or dandruff associated fungi or bacteria, but actually the proportion of those bacteria and fungi varies in the healthy and disease condition. So but to get to reach to this kind of conclusion, you have to carry out a large scale study on different individuals, get the data, do the analysis, and then you should also look at this data in connection with the other omics data also that you may get, and then you can tell a better story or a better picture of that disease complexity. Okay, then you can also see the impact of various external things that we apply on the skin or scalp, and how they can influence the microbiome or the health of the host. Then particularly this gut microbiome has been associated with diabetes mellitus, which is a disease of basically associated with like eating also, and it could, people are predisposed to this, or it could be genetic, or you are acquiring it later, and basically it is because of multiple factors, environment, your eating habits, physical lifestyle, and many other things, right? So age is one of the factor, uses of excess antibiotics, diet, lifestyle, age. Okay, you can have a genetic background, the delivery procedure, okay, bacteria in the amniotic fluid, breastfeeding, and lot of things can influence this, because actually this gut microbiome is playing a lot of role in the amino acid synthesis, dietary fiber absorption, fat soluble vitamins, okay, scfas and bca, branch chain amino acids and short chain fatty acids. Okay, so the healthy microbiome will do all these jobs, protective functions, metabolic functions, structural functions. But when this is diseased, or basically in the dysbiotic state, it can lead to diabetes, and people have found strong association of the altered gut microbiome with the diabetes mellitus, and which is typically basically a metabolic disease characterized by pancreatic beta cell dysfunction and peripheral insulin resistance that lead to defects in glucose metabolism and chronic low grade inflammation. So basically, along with the genetic, there could be several other factors, other environmental, calorie intake, nutrient composition, ambient air pollution, physical inactivity, which can lead to diabetes. But when we are assessing it, the role of microbiome and is also very equally important. And to assess the diabetic condition, you will have to look at the uh, SNPs or the GWS studies data, the metabolomics data, metagenomics data, transcriptomics data. Okay, so this is one of the example diabetes where you can use multiomics approach to understand the disease, its progression, its response to various therapeutics, and also early diagnosis and like that. Okay, so you can take like obese or diabetic patients, because obese are also predisposed, or basically are at a high risk for diabetes, right? So, and then basically people have also found various type of uh, uh, firmicutes to bacteroides ratio, or association of particular bacteria such as lactobacillus to be very high in the diabetic compared to non-diabetic, and like that. So when you are studying transcriptome, genome, um, metagenome, so metabolome also becomes equally important to see the actual final, because metabolites are the final byproducts of the metabolic processes, and that can provide evidence about what has happened, or what hypothesis you have given about the role of a bacteria or a particular enzyme, by detecting that metabolite or its derivatives in the various kind of, it could be tissue or could be an excreta, or various biological fluids. So that is why metabolome or is one of the important component of the overall transcript of the omics approach in understanding the various diseases, such as it could be cancer, cardiovascular, or diabetes. Okay, and people have shown that there could be metabolomics can be done by several means, like mass spectrophotometry, MS, it could be GC or LC or NMR data. So people have tried to connect like cancers with various kinds of analytical approaches, and showed that these metabolites can be used as markers or provide early indications for the existence of cancers. So to do this kind of analysis, basically first of all we have to understand what the metabolome is, and where it is and how this can be used. So basically metabolome is can be defined as the complete complement of all small molecules less than 1500 daltons, and in a specific cell, organ, or organism. So basically this data or metabolomics is used along with the genomic, transcriptomic, or proteomic approaches, and together they can constitute the overall systems biology approach for to understand the system. And metabolome is a recent field, evolving field, though we have a lot of data in the human genome, transcriptome, proteome, but in metabolome data is coming up, and still lagging behind the other omes. And for this, basically one of the best resource to get this kind of data is the human metabolome database, which contains huge repositories of the human metabolites and their sources, respective cell types for the different populations studies, right? And basically it contains more than 2000 endogenous metabolites which are present in the cell, and also provides you a server for your analysis. You can upload your metabolomics data because it has the data at its backend, which can be used as a reference data, and then you can compare the peaks and provide you the analysis output. So basically HMDB has emerged as the one of the best resources for the human metabolome, which is connected to almost 115 non-redundant pathways, almost 2000 distinct enzymes, more than 1 lakh SNPs, and 862 metabolic diseases, okay, genetic and acquired. Okay, more than 400 compounds have been linked to experimentally acquired reference by the various NMR 1 or 13, 1S13C or GCMS data. Okay, then comparative study with various healthy and diseased, various tissues, urine and other biofluids. Okay, so basically it's a very exhaustive resource of the metabolites that have been extracted from the human body sites, and it's a very comprehensive data which you can also download for your analysis and also use it online. And similarly, like epigenome, as we have talked earlier in the previous lecture also, is one of the important aspect which should also be studied along with the other omics, because it is the methylation pattern of the genome, right, of these CPG islands or the Cs in the genome or the histone modifications, which may impact the expression of genes, which ultimately are the functional moieties and play a very important role in the health and disease of an organism. So epigenetics, metabolism, health, disease, diabetics, very rare disease, metabolic syndrome, all have been connected. Epigenetic profiles can change based on the environmental exposures, diet, a lot of factors like pollution, environment, right, diet, exercise, all these things can influence your epigenomic profiles, and which can lead to epigenetic modifications and can play or can be associated with various health and disease aspects. And basically, like just to give you an overview, it is the methylation of the cytosine, which may be present upstream or downstream of the control sites and can lead to this kind of their variation can, this epigenetic changes or modifications can lead to down expression or more expression, or even lack of expression for particular gene, which can be associated later with the disease component. Like you can see in this picture, various C's and G's have been highlighted in this, and this is the start codon. So you can see the like it is a possibility that many of these CPG may get methylated, and that also may be differential in among different states and individuals, and that can lead to that can be associated with the various diseases. And people have found the epigenetic marks in normal and cancer cells, and these changes in the transcription start sites or hypermethylation imprinting, or in the repetitive sequences, the hypermethylation. So those have been associated with as marks or markers for the of cancer, and along with the histone modifications such as acetylation or methyl or phosphorylation of the histones. So that is why the Human Epigenome Project generated the catalog of all the genome-wide DNA methylation patterns in all the human genes in all the major sites, and people can use this as the reference database for the comparative analysis, and which can provide first-hand information and growing information on the methylation states of various tissues and in terms of health and disease. So basically, the future of cancer epigenomics is it can provide markers for the various cancer type of cancer. You can get epigenetic modification which can be associated with various type of cancers, help in tumor progress, prognosis, predicting the various chemotherapy or drug responses, and also like re-expression of the silence genes, therapeutic uses, and creating the human epigenome. So this field along with the other omics is very important again to understand the disease pathogenesis and its cure. And people have done the like the GWS epigenome wide association studies also, where in the cases and controls you can for a particular tissue they can look for the genome wide epigenetic maps, and also along with that the GWS and the SNP maps, okay, and can find the epigenomic variations in the control and healthy individuals, which can provide insights and also leads for the experimental validations and further studies. But to confirm these you need a lot of data, and you have to establish it in multiple individuals, do the statistical validation to basically find something or tell that okay, this kind of epigenome wide association or this epigenome based association is associated with that disease. Similarly, for the cancer, there is a cancer genome atlas, which there are so many types of cancer. It also varies according to age, lifestyle, population. So researchers have made the cancer genome database, which is Genome Atlas program, basically TCGA, which is a landmark cancer program for like over 2,000 primary cancer to 20,000 primary cancers spanning like 33 different cancer types, and provides data about the cancer, their genome-wide association studies, metabolomes, expression profiles, and this is very important to understand cancer genomics and to understand cancer. And basically this atlas is one of the major resource that you can use for obtaining various kinds of aberrations in DNA sequence, gene expression, epigenetics, the regulatory RNAs, protein expression, structural variations in the tissues. Okay, so basically the lot of comparative data from healthy and disease is available here. And just to give you the insight, like it contains about 2.5 petabytes of data from 33 different tumor types, including 10 rare cancers, from more than 11,000 patients and 7 different data types, and can provide the molecular basis of cancer, tumor subtypes, therapeutic targets, and collaborating like almost 20 institutions across the United States and Canada, and also getting data from other places. So this is a very exhaustive database for cancer research. Now in the end, we should also understand, or basically just to give you an overview, that once you are doing these kind of metabolomics approaches, the use of animal models is also like very important to establish or to show that in animal model, and also for going for the clinical trials. So basically once you have formed the hypothesis in the Petri plates or in a given set of data, you would like to use the mice for the hypothesis validation, and rodent animals, and go for larger animals for the clinical trials, and later on you will go for the clinical studies. That also is in different phases, and only then the outcome of various omic studies, the leads or the proof of concept that you have made there, you will be validating using the clinical trials in various model systems, and then you can convert your proof of concept into a transnational product which can come to the market, and it could be a therapeutic, diagnostic, or any kind of offering from your research. So in this lecture we covered about we talked about the different types of multiomics approaches that can be used to understand the diseases, and like how it can help you differentiate between health and disease, and also in terms of markers, and how the multiomics approach can be used here, such as in the cases of cancer, diabetes with examples. So this is good for this lecture. Thank you.